or glucose carbon substituted with heavy 13C isotopes, and then based on the positional enrichment as well as the number of carbon substitutions in their positions, we can actually determine what type of biochemical reaction did participate in producing the intermediates that we were interested in studying. And more specifically, this is how this glucose molecule looks like. There are two 13C carbons in the first and second position. And the reason using this carbon design in glucose is because it's the most effective in studying metabolism. And actually there's a recent paper from the Massachusetts Institute of Technology for participants who are not familiar with what MIT is. It's a Boston-based private research facility, and they are the ones who sent people on the moon. They are certainly regarded as the one of the most prestigious research institutes in the United States. And actually they have reported just very recently, citing our papers, that this tracer has the highest precision scoring in metabolomics type of studies. The experimental design included incubation with the tracer. I'm going to present data at the 72 hours time point. We looked at cell pellets and media, and just as I described, we followed this scene or scenario for the studies. The deuterium depleted water did not, and this is, I'm jumping to the data so we can talk about some specifics in more detail. We did not see any difference as far as glucose uptake or complete oxidation or glycogen synthesis in any of the cell lines. And I'm going to show you just what we found as far as glucose consumption. This is a milligram per 72 hours and by million cells. And as we look at the differences in the pancreatic cancer as well as the lung cancer cell line or the breast cancer cell lines, we did not see any significant change as far as glucose uptake. And obviously this is a 3-culture study, so the standard deviations are somewhat high in some of the groups, but this is a pilot study, so we were just trying to understand the effect of 150, 150, and 25 ppm water on glucose metabolism. But anyways, there was no significant uptake, increase or decrease in glucose uptake. Now, interestingly, when we looked at the pentocycle flux relative to glycolysis, it was decreased in one of the cell lines. I'd like to point out that there's only one common metabolic effect to be found, and I'm going to talk about fatty acid synthesis later, but yet the cell lines responded somewhat differently to deuterium depletion whenever they responded. And this is a function, or this is a functionality that would tell you how much NADPH or how efficiently NADPH is produced in these cell lines, how effective the oxidative branch of the pentocycle is. And as you can see, there was a significant change only in the decrease after all deuterium depletion scenarios in mesopancreatic adenocarcinoma cell lines, while the other 2 cell lines did not respond really to deuterium depletion as far as oxidative ribose synthesis versus glycolysis, glucose going through glycolysis. And the MCF7 breast cancer cell line was actually with the 125 ppm water, it showed an increase in this study. Size did not show any significant values, yet there was a trend. Now, this number on the y-axis tells you exactly of every glucose molecule that enters the cell, how many of them went through the oxidative branch of the pentocycle and got returned into glycolysis through the non-oxidative branch of the pentocycle. which is 5 out of 100, between 5 to 6 out of 100, meaning that 5%, 5 to 6% of glucose goes through the oxidative branch, which was decreased significantly in MIA cell lines. And the technology works in a certain way as glucose enters. As I told you, there's 2 carbons labeled in the 1 and 2 position, and glycolysis will process this glucose toward glycolysis, through glycolysis towards lactate. And in the meantime, there is a loss of carbon from the first position of glucose, and ribose is formed, which is after formation, it's recycled into through these enzymes, which are transkylase, transadolase, into glycolysis, and then the process goes very similar as far as high glucose or the 6-carbon product, fructose, is broken down into lactate. But what's really interesting is that lactate that is produced through glycolysis directly is going to have 2 carbon substitutions in the second and third carbon of lactate in the plasma or in the media, while glucose that goes through the oxidative branch of the pentose cycle and goes through glycolysis, it will have only 1 carbon substitution. So using mass spectrometry and GCMS and gas chromatography, we can always accurately determine what is the fractional synthesis of lactate through these various patterns, more specifically through the oxidative branch of the pentose cycle. And according to our results, which is very common in tumor cells, about 5 to 6% goes through some tumor cells. In liver cells, it's much higher, about 15-20% of glucose goes through the oxidative branch of the pentose cycle, and there's a tissue phenotype, kind of marker value of this lactate based on the carbon substitutions, and which reflects on the glycolysis versus pentose cycle flux. But anyways, this is the reaction, more specifically this NADPH producing reaction, and this hydrogen is used in, this comes from water, and this hydrogen is used for de novo fatty acid synthesis. Now, looking at RNA ribose synthesis for nucleic acid or ribonucleic acid production, which is, again, it's a different story as lactate production, but RNA ribose would be generated from glucose in the media. And the 25 ppm water decreased this value in the MIA pancreatic adenocarcinoma cell line, very consistent as what we would see with the lactate producing pattern, but in the H441 and the MCF7 breast cancer cell lines, there was no difference. So this indicates, again, there is a tumor cell specific response to deuterium depletion, and it seems that the pancreatic cancer cell line, the MIA cell line, is responding to deuterium depletion through the oxidative branch of the pentose cycle. So again, for ribonucleic acid synthesis, this is our glucose that comes in, and through the oxidative branch there is a carbon loss, and through the non-oxidative branch there is a preserved carbon configuration in the ribose, and this actually, based on where the substitutions are, we can differentiate among the oxidative versus non-oxidative transkylase, transadolase. This particularly is transkylase, which actually transfers a keto group from top of fructose onto glyceraldehyde 3-phosphate. Obviously, if there is triose phosphate isomerase activity, then the bottom 2 carbons can get labeled in glyceraldehyde 3-phosphate. So there's a number of scenarios which will be disposed in this particular scenario, but for this study we were actually looking at the total labeling and more specifically how much glucose is accumulating or ribose is accumulating through the oxidative branch of the pentose cycle, and that was inhibited by deuterium depletion. Now, the TCA cycle substrate flux is, again, it's a very important measure of energy production and cell survival, and that seemed to be more responsive in the MCF7 breast cancer cell line. And what you can see here is that, in particular with the 25 ppm water, the labeling in glutamate, which is actually the product of pyruvate entering the TCA cycle either through pyruvate carboxylase or pyruvate dehydrogenase, eventually through citrate and isocitrate formation, the certain labeling scenario in glutamate would allow us to determine a number of things. And one is how many times the cycle turned over, how effective this